Hey, Bio. All right, so we're going to do a quick little extra help review video here, just so you guys are prepped and set up and ready to go for the test. So I'm going to jot a couple of things down here on the board, and I'll switch back and forth a little bit between my computer and what's on the board here. So first off, we're talking about RNA, right? And again, we know RNA. We just kind of went through this. It's single-stranded, right? So unlike DNA, right? Which has a double helix. We know that DNA's got, the double helix. We know that it has deoxyribose. We know that it uses A, T, C, and G. It does have base pairing. Okay, and that's coming together to make it very stable, right? RNA we know is single-stranded. Okay. We know that it's got ribose as the main sugar. It's got AUCG. There's limited or no base pairing. Okay, so that's real important to know. And the other thing is RNA is a complementary copy of the DNA. So remember, it's an opposite copy, right, of the DNA. That's really important because DNA, right, when we copy one strand of the DNA, we're going to copy it and read it 3' to 5'. The new strand of RNA is always 5 to 3, right? That's its orientation, right? So the strand that you make is always 5', 3', okay, for RNA. That's for any type of RNA because RNA is a complementary copy of DNA, okay? So what's real important is you guys know the 5 types of RNA, okay? Because RNA has lots of different functions, okay? I'm going to just make like a little circle here because this here is really a unique group of RNAs. We have the pre-RNA, okay, which is newly transcribed. That we copy from DNA. And then this needs to get processed, right? And we have snRNA, right? This is going to form spliceosomes. And the job of this is to splice out / remove the introns, right? So this is going to take those introns out, and it's going to put together the coding sections, which are exons, okay? This is only in eukaryotes. That's why I did this like this. And this is only happening in the nucleus, okay? That's where this occurs, in the nucleus, okay? Once you splice your RNA, now you have messenger RNA, right? Which is a copy of one gene, okay? And it codes for a polypeptide, right? So it's one gene copy that's coding for one polypeptide, okay? That's your messenger RNA, right? The messenger RNA gets read by rRNA, right? Which is your ribosomal RNA. Okay, and that ribosomal RNA, okay, that's going to read your mRNA gene sequence, okay? And this is going to assemble proteins, right? How do you make proteins? You put together amino acids. So what do you need to bring amino acids to this? Your tRNA, right? That's going to carry amino acids, right, to the ribosome so you can put those amino acids in the right sequence, right, and create a protein, okay? So those are your 5 types of RNA. There are a couple others which we'll talk about in the next chapter. But pre-RNA and snRNA, those are found in the nucleus in eukaryotes. So these are eukaryotes only, okay? So when you first copy the DNA and transcribe it in eukaryotic cells, it's transcribed into pre-RNA because this still has introns in it, okay? And then what happens is these snRNA molecules form spliceosomes, which go in and cut out the introns, okay? And it gets rid of the introns so that what you're left with is a mature piece of messenger RNA that doesn't have any intron, okay? Then this goes out, goes to the ribosome where it gets read, and it's a copy of one gene. Remember, this is a complementary copy of one gene. This codes for a polypeptide, and that's going to allow you to make protein, okay? A couple of things now as far as transcription, translation, and all that stuff. So let me erase this, okay? Okay, remember transcription that's DNA to RNA, right? So you're going to copy one side of the DNA and make a complementary copy of RNA, okay? This is in your 5' to 3' direction, okay? Translation. This is where RNA, specifically mRNA, is read by a ribosome to make a polypeptide, okay? So translation is where we take our genetic code sequence and convert it into a polypeptide sequence, right? Putting together amino acids, okay? So that's the main difference between those two, okay? So the central dogma of DNA basically is you have DNA to RNA to protein, right? That's the central dogma, okay? That's the flow of genetic information, okay? Goes from DNA to RNA to protein, all right? Now, transcription. Okay, let me show you a quick shot here of transcription. So let me put my screen up so you guys can see. And when you transcribe bacterial chromosomes, right? In a bacteria, this all happens in the nucleus. I'm sorry, in the cytoplasm, okay? There's no nucleus in a bacteria. So here's our bacterial DNA, okay? So it's a circular chromosome. We have one little section that we're going to open up. That's a gene that the bacteria is interested in expressing, right? So when you express a gene, what you do is you transcribe it into messenger RNA, and then that messenger RNA gets translated at the ribosome into a protein. Okay, so there's your flow of genetic information, all right? Now, in a eukaryote, this goes a little differently because we have to process, right? Remember, there are three things you have to do for RNA processing. You have to add a poly A tail, a methyl cap, and then you splice, right? And the idea is that by splicing out, you're taking away those introns and you're combining the exons to create your mature mRNA transcript, okay? So here's our methyl cap that we add, right? There's our poly A tail, okay? And when we do this, right, methyl cap, poly A tail, that's protecting the main gene here, which is this messenger RNA, right? So this is the information is in here. The genetic sequence in here codes for the protein, okay? The snRNA forms these little things called SNRPs, right? A SNRP is consisting of 4 of these little snRNA molecules, okay? And that's going to help pull these introns inward and create a little loop, and it pinches them out. And what you're left with are the exons, which get put together, okay? In eukaryotic cells, transcription and the processing all happens in the nucleus, right? So this is your pre-RNA, your primary RNA transcript, and this is your mature mRNA after it's gone through the processing. The introns get pulled out by the spliceosomes, okay? And the green areas here, that's exon, right? That's what codes for protein. Once you put that cap and tail on, that protects the messenger RNA so that it can actually leave the nucleus through a nucleopore and make its way out into the cytoplasm, where you can safely translate it into protein. Okay, and you can see the difference here between your DNA and your final mature RNA after it's been spliced. All of this green area here represents exon, which is coding for protein. Okay, and then here's our cap and our tail. All right, so that's transcription in prokaryotes and in eukaryotes. Okay, the next piece, which I want to get to, and let me just put myself back on the camera here so you guys can see me. You don't want to stop sharing that. Okay, great, we're back on. Is what happens when you get that ribosome involved, right? So here is your messenger RNA. okay? And your messenger RNA has, in a eukaryote, has left the nucleus, right? We've sliced it, we've sliced and diced it, we've done everything we need to do, and now this is ready to be translated, right? And so the first thing that happens is it's going to bind to the small ribosomal subunit, okay? Which is going to attach to your start codon, right? Remember, each of these codons codes for an amino acid, except for that last one, right? So then what happens is your first tRNA molecule is going to orient itself, right, and pair up. Here's your anticodon, right? So you have an anticodon here. And then up top here, that's your amino acid, okay? And the tRNAs are carrying the amino acids, right, to this ribosome. Now, ultimately, what will happen next is your large subunit is going to attach, okay? And this is made of rRNA, and this reads your mRNA strand, okay? And what this will do is move along your messenger RNA strand. And each time it moves along, it's going to move one codon over, right? That's translocation. Translocation, changing its spot, changing its position. And it's going to continue doing that until you hit the stop codon. How to find another amino acid. Okay. So now, how does this all work? Okay, so this is your ribosome, okay? And so your ribosome now has this large subunit that's going to attach, okay? And remember, this large subunit's got 3 spots to it, okay? It's got the P site, the A site, and the E site, okay? EPA, like Environmental Protection Agency, okay? So this codon here is GGC, okay? So we're going to look for that next tRNA to come in, okay? This is going to come in here. It's going to align itself, okay? And then what happens here is the P site catalyzes a peptide bond between these 2 amino acids. So this methionine and this glycine are now going to connect, okay? This whole ribosome is going to shift over one, right? When this ribosome shifts over one, this now is in the E site, this is in the P site, and the A site's open. And then that's ready for the next tRNA to come in, okay? And that next tRNA is going to be this valine, which comes in, okay? And it pairs it up. This goes into the E site, and then it exits. And what does it do? It goes and it picks up another amino acid, right? And then your methionine and glycine are connected, right? This whole thing shifts over one, okay? And now you're set up like this, okay? And what's happening is your ribosome is moving codon by codon. And it's going to continue doing that until you hit the stop codon. Remember, the stop codon does not code for anything, right? Stop codon codes for stop. There are no amino acids that code for it, okay? I have the link to this video sending out on Remind at about 3 o'clock today. So you'll be able to see that. You'll actually probably get the video before you get this. So you'll have already watched it, and then you're seeing this again. That's how it works. But the ribosome is going to continue to read this, right, until you get to the end, right? And then at the end, you have that stop codon, and everything dissociation falls apart, okay? Now, sometimes mutations happen, right? As the ribosome is reading, it's very possible that it could skip a base. So it may start off AUG, GGC, then it skips this A, right? And it reads this as GUG and UCU. And that's a problem now because that frame shift now has created 2 new codons that didn't exist before because you're skipping this, right? And that results in totally new amino acids, right? Which means now you have a completely different structure to your protein, okay? So by changing one of the bases, you're going to most likely change one of the amino acids, okay? Or more, and that results in a major mutation, okay? Now, if you just change one base, if you take like this G and swap it out for a C, that's a point mutation, right? That's happening at one point. It's a discrete mutation at one spot on the RNA. But if you remove a base, now everything shifts over. So if we took this G out completely, now this is going to be AUG instead of AGU. Okay, if you take this out, and everything is shifted off, and now you have a whole different type of amino acid sequence than what you had before. Okay, so those are some of the problems that can happen. On the test, you will be asked, there will be some questions that ask you, what would be the result of swapping out one of these bases, right? And you have to look and see, does it change the amino acid or not? Usually because the code is degenerate, which means you have more than one codon coding for each amino acid, that's going to reduce mutations, okay? Because the chances of swapping this U out with an A, pretty good, but AGU and AGA probably will code for the same thing. I don't have to look at the chart and see. But it's usually that third base that if that changes, you're going to wind up with no real effect happening because more than one codon codes for each amino acid, and the changes happen on that third codon, so everything's still the same. I'll show you guys what I mean. Let me put the chart up here. Let's see, where is it? Here we go. So again, right here, okay, here's your chart, and you can see that, for example, leucine has 6 different codons that code for it. We have a UUA and a UUG, and then we also have CUU, CUC, CUA, and CUG, all code for leucine. The first 2 bases are the same. It's the third one that has the high variation, okay? So like leucine has CU for all of its first 2 bases, and then the last base, the third base, is the one that's most likely to change, okay? That's called degenerate because you have multiple codons coding for the same amino acid, okay? You also have 3 codons that don't code for anything. Those are your stop codons, okay? They just indicate that it's the end of the gene, okay? And MET is your start codon. So remember that when you're copying DNA into RNA, when you're transcribing it into RNA, you're always going to start transcription at a start codon. Okay, so the difference between transcription and DNA replication is DNA replication just starts anywhere and you copy the whole strand. With transcription, you're copying the DNA only between the start and the stop codons. Okay, so that's real important to remember that when you're looking at different questions. Okay, let's see. So we've just hit the differences between DNA and RNA. We did the 5 types of RNA. Really important. Make sure you guys know the 5 types of RNA. Not just because you're going to have questions that ask you, which type of RNA is snRNA, what does tRNA do? But if you know the functions of the 5 types of RNA, that almost automatically helps you understand the whole process of protein synthesis, right? Which is translation. Because you've got your tRNAs here, rRNA is a ribosome, here's your mRNA, okay? Remember that it's a complementary copy. So this is a 5' to 3' copy of your DNA template, okay? That's really it. That's everything that I think you definitely would need to know for the test. Let me just take a quick peek back through the notes here and just make sure you got transcription, translation, 5 types of RNA, right? Make sure you know your RNA structure. Splicing, that's really big, okay? That eukaryotes have the pre-RNA, which then gets spliced out, and you're going to remove the introns, and you're going to add the poly A tail and the 5' methyl cap. That's real important there, okay? Definitely make sure you guys know protein synthesis, okay? The whole thing I just put up here on the board, okay? That's it. If you have any questions, you can reach out to me on Remind. Again, AP Classroom is up and open. Please make sure you guys do that. AP Classroom is going to be graded, but it's going to be graded as a completion grade. So you don't have to do the, you don't have to worry about me like putting in like a 50 or a 60. It's going to be, out of whatever points I decided to be, right? So just the fact that you did it, and that's it. You also have the answers to the 2 worksheets that we did today and yesterday in class. So I posted those up on Schoology already. Not grading them, but they're definitely going to help you for the test. So it's a great way to review, okay? And that's it. So good luck, guys, and I'm looking forward to seeing you guys tomorrow. And good luck studying. Have a great night. I'll see you tomorrow. Bye.